Hi, my name is Jessica Kern. I'm the laboratory manager for the Ecology and Biogeochemistry Core Laboratory. The mission of the laboratory is to look at carbon, nitrogen, phosphorus, and hydrogen cycles in plants, water, soil, and snow samples and develop cutting-edge research methods for these procedures and engage researchers from all over the nation. I put together a faculty steering committee with 7 researchers across the University of Wyoming campus from all different departments. They help me with the direction of the laboratory to assist in the further expansion and research needs. Hi, I'm Jessica Kern, the manager for the Ecology BGC laboratory here at the University of Wyoming, and I wanted to take you into the laboratory. Here is the lab. You will see to the right we have some of the shared instrumentation, the ion chromatograph, the elemental analyzer, the fusion for TOC IC. In this room we have a couple different centrifuges, an extraction robot, and the freeze dryer. In this room there are 4 shared ovens at varying temperatures, and then we have the automated solvent extractor and a pulverizer. It's used for rock and soil samples. We have some shared balances, and then in here we have a hydrofluoric acid hood and another chemical safety hood. The ultra pure water system is here. Identifying the needs for the laboratory, 4 large equipment purchases were made, and these were available in June 2019. I've highlighted some of the major analytical capabilities of the Ecology BGC laboratory, and you can see the different sample matrices that we are able to test. Busy developing standard operating procedures for the analytical capabilities and field collection procedures. There is also a tablet that will be in the laboratory that has videos for students and researchers to follow.
I'm using a 2mm sieve to collect my rocks because this is the type of soil we want to use for our analysis in the lab.
Just want to say that my method requires the oven to go up to 85 degrees, and here you can see the set temperature and what the actual temperature is right now, and the instrument will wait until the temperature reaches 85 degrees or whatever temperature you have, and it says here oven wait.
Our lab is open to any researcher and student to use to process their samples. The lab received a small micro grant, and this was used to purchase 3 field kits for lab portability for outreach activities and researchers' needs. These kits can be used to test plant, soil, water, snow samples in the field. The lab helped with 2 internships, SHRAP and Hard of Hearing. These internships gave high school and college-age students the real-life experience of field collection, laboratory experience, and presenting in front of an audience. I used the help of one of the hard of hearing students who had expertise in CAD software to help me design a new filtration rack for the laboratory. This filtration rack now has a design patent on it, and I used the UW Technology Transfer and Research Product Center to help me with this. Core laboratories were able to hire 8 undergraduate students. The students were instrumental in helping both laboratories get set up this first year. The lab had a large impact this past year, running thousands of analytical samples, not only with researchers involved in the grant, but those at the University of Wyoming campus. The lab also hosted over 250 elementary age students across the state of Wyoming. I am always looking at adding new analytical capabilities for the lab. The next one that I'm going to be adding is the phospholipid fatty acid analysis for microbial biomass and fungal to bacteria ratio. I will be using a gas chromatograph for this, as well as adding other analytical capabilities for the other instruments. The lab has been affected by COVID-19 and the CDC guidelines. I've had to restrict access to the laboratory as well as taking more time to process samples and cleaning and sterilizing.
Hi there, this is Alex Berkeley, and I want to talk to you about a project-related facility, in this case, the Genome Technologies Lab. I've spoken to you in some of the other videos, and in this case, I'm speaking to you as the director of this facility. This facility is a brand new facility that was initiated at the university as a result of this project, and is staffed by Greg Randolph, a research scientist who's been part of the project since early on, and has helped us build this from scratch and equip an empty laboratory with equipment and technology to support our project and other projects on campus. Our goal for this laboratory is to produce economical high throughput library preps. And these are sequencing library preps, either whole genome sequencing library preps, or for this project in particular, the largest number are amplicon library preps that would then be sequenced at other facilities. So we've not equipped this lab to be a sequencing lab per se, in that we have not acquired the really top shelf expensive sequencing instruments for ourselves, but instead we go to core labs elsewhere to use their sequencing equipment, either commercial labs or laboratories at other universities. So instead, what we've focused on is liquid handling and methods for us to do the library preps from all the way from extraction all the way to completed library that's ready to submission. And so to do that economically and in a high throughput and repeatable way, we've acquired some robotics. And so in the first case here on the left, there's an Integra robot that's exciting because it allows us to move liquid from 2 milliliter tubes into 96 well format plates, and which is a critical component of going from field sample into either columns or other type of formats that are used in extractions. In the center, we have a robot that we've just acquired, a pipetter with 96 96 channels, so that we can stamp out and add liquid to every one of the wells in a plate very rapidly. And then on the far right, we have a Hamilton robot that's in an enclosure that allows us to do a variety of different things. We do dilutions after quantifying our DNA and other settings, because that's the pipetter in that machine has 8 independent channels that can pipette different volumes independently of one another. And so it's very high throughput for doing normalizations, for example. This is also where we set up our PCRs or do complicated liquid handling associated with setting up our arrays of oligonucleotides for amplifications. We also have a variety of facility equipment associated with quality control and assurance. And so on the left here, we have a real-time PCR machine that we acquired. Actually, it was equipment that was purchased for a much earlier EPSCoR grant, but this machine is still very usable, and so we're able to move it into our laboratory to get a good idea of the amount of product that's in our samples, and to assure that it's amplifiable for and sequenceable in an Illumina sequencing setup. We also have the spectrophotometer that is very flexible, but we typically use to quantitate our product and then transfer those quantities, those concentrations to the Hamilton robot to do our high throughput normalizations as needed. Additionally, we have this bioanalyzer to look at traces of the molecular weight of our sequencing libraries, as a sequencing core lab would do. This way we can make sure that we have products of the expected size and nothing of unexpected size in the library that were submitted. And we did acquire an Illumina iSeq instrument, the small cube there, white cube next to the freezer with a screen, that is a Illumina sequencer that is relatively inexpensive and allows us to do proof of concept sequencing or very small scale projects on site without needing to send them off. So in our project, because we have taken, or because we're being so ambitious about the number of samples that we're looking at, we've really taken a look at every single step of the methods that we're utilizing or that we're using. Everything from extraction to library preps to sequencing itself and the bioinformatics and analyses, and looked for ways in which we could innovate to make the most of the funding that we have available and the sampling capacity. And so we've innovated in terms of internal controls that we have, in both in terms of being able to convert from compositional data to absolute abundances. We have internal controls with respect to sequence identity or sample identity to guard against contamination or carryover of sample from one to another in our library preps. On sequencing, we really, because we've designed our own arrays of oligos, we can go at very high multiplex and use NovaSeq instruments and very large numbers of reads split across many samples and many loci. And then we've designed a bioinformatics workflow and statistical analysis to make good use of all of those sequence data. So we multiplex up to 9,216 samples in a library, and potentially would include 2 loci, and that's what we've done in our first 2 libraries that we've sequenced, where we've sequenced 2x250 base pair libraries on a NovaSeq. And 2 of those libraries have been sequenced already. 1 is currently being sequenced, and we've completed 7,005 unique metagenomic samples to date. One more library is near completion, or one more project, which will take us all the way through all the samples that we had collected up through this summer of 2020. And we're getting large numbers of reads per sample and locus in those libraries, and the variance is in part by the number of samples that were loaded there. But we have plenty of information with which to draw inferences about the frequency and relative abundance of taxa in those samples. As you've heard in other presentations, you heard about the seed grants potentially already. In those, we made an award for a group to do eDNA work in freshwater systems, and so we've adapted our multiplex protocol to work with CO1 cytochrome oxidase 1 for amplicon detection of animal taxa and community composition in freshwater samples. We've also in the last months adopted this multiplex method as a next-gen sequencing method to analyze amplicons from N1, N2, and RNase P, which are the 3 standard loci used to detect the coronavirus. And we'll evaluate in the coming weeks whether we will potentially use that for surveillance in Wyoming. Overall, we've worked with the university to seek sustained funding for our staff scientists and the hourly assistants who work with us. And so we are in the budget for the Office of Research and Economic Development for the period starting after the grant. And overall, we're working with the university to work within the standard budget model and accounting system and operating procedures to have this be a core lab that's sustained by the university. Thanks much.
Hello, my name is Franco Basile. I am an analytical chemist at the University of Wyoming Department of Chemistry. I'm also a member of the science team of the Microbial Ecology Collaborate project. Our lab is focused on development of thrust 2 of the proposal, which measures the microbial life functional consequences. My lab uses mass spectrometry as the primary analytical tool, and with that we measure the proteome or proteins and the metabolome or metabolites present in a sample. I will next highlight 2 projects associated with thrust 2 of this proposal. The first project we are measuring the proteome or metaproteome in this case of this ecosystem. The ecosystem involves cheatgrass, an invasive species that's held at bay by wheatgrass. And so our measurements are looking at the functional aspects of the microbial life associated with the soil samples with wheatgrass and cheatgrass. This project is in collaboration with Professor Linda Van Diepen in the Soil Science Department, and grad students Brandon Says in my lab is working on the proteomics aspect, and Gordon Custer is a grad student in Van Diepen's lab. In addition to the mass spectral measurements involved with the metaproteomics analysis, there is a large bioinformatics component of the metaproteomics project, and for that we're teaming up with members of the bioinformatics core at the University of Wyoming, also with Joshua Harrison, a postdoc in Alex Berker's lab, and the Galaxy P team at the University of Minnesota. Combined, we expect to extract and obtain information about the functional state of the microbial life associated with samples in the wheatgrass and the cheatgrass soils. The metabolome project that is currently in progress in my lab is in collaboration with Cynthia Waning's lab, and involves the measurement of root exudates under circadian rhythm conditions. The project is being carried out by Mitchell Helling, a postdoc in my laboratory, and Alessandro Ceretto, a grad student in Cynthia's lab. And so they've developed a method to use dialysis probes to sample in situ the metabolites or root exudates, and then these are analyzed by LC-MSMS. And right now we have very preliminary data on this setup. Now, any of these projects would not be possible without the acquisition of a state-of-the-art liquid chromatograph tandem mass spec system with funds from this Track 1 grant. And this is an incredible resource that has expanded the research capabilities at the University of Wyoming, and also enable us to train undergrads, grad students, and postdocs in highly sophisticated analytical and it has also expanded our capability to engage in a wide range of research projects.
My name is David Williams. I am a professor in botany here at the University of Wyoming, and I'm the faculty director of the core facility, stable isotope facility, that is administered within the Office of Research and Economic Development. The facility was founded in 2004 and has benefited immensely from awards from the National Science Foundation. We currently have 2 permanent full-time staff. You can see the pictures at the bottom, as well as a number of part-time technicians, including undergraduates who work in the lab. I'm the PI, or the faculty director of the facility, but responsibilities for overseeing the facility are shared by faculty from across a number of departments on campus. We currently have about $1.5 million in equipment assets within the lab, and from recovery of costs from doing analyses for our collaborators and researchers, our annual revenue is about $150,000 to $200,000 per year. What we do sample analyses supporting research in numerous disciplines, a number of departments across the University of Wyoming. About 50% of our work is for researchers at the University of Wyoming. The other half is from external researchers that are at other universities within the U.S. or internationally. One of our largest contracts in the lab now is with the National Science Foundation NEON project. We're also involved quite a bit with education, outreach, and service. For example, I teach a graduate stable isotope ecology class every other year. So for the current project in microbial ecology, our lab is supporting work to trace nutrient sources within microbial communities and ecosystems. These techniques are really important, allowing us to quantify biogeochemical transformations and fluxes. And importantly, these techniques allow us to detect the presence and record the history of microbial metabolisms within Wyoming's wild ecosystem. This slide shows the mass spectrometer system that we've acquired with the current EPSCoR award. What's unique about this system are the two peripheral devices on the left-hand side that allow us to do some pretty interesting things. The elemental analyzer in the bottom left has some unique capabilities. It allows us to measure the carbon, nitrogen, and sulfur isotopic composition, as well as their elemental abundances on single samples. And that's quite important and useful for our current EPSCoR project. And just to give you a perspective of the capabilities our lab has relative to those in other states around Wyoming, none of these capabilities are available in Colorado, Montana, Idaho, Nebraska, North and South Dakota, or Kansas. And there are only two other labs in the region that are capable of doing these kinds of analyses. One is at the University of Utah, Turi Serling and Jim Ellringer's lab, and another is Zach Sharp's and Seth Newson's lab at University of New Mexico. These are fairly internationally renowned laboratories, and so we're in pretty good company. So the goals for the next year are to develop a new method for getting at and converting nitrate to nitrous oxide. The current method we have is quite laborious, and so we want to develop a new method. And finally, to further refine some of the tricky parts of doing sulfur isotope analysis on plants, soil, and animal samples. Thank you.
This portion of the presentation, we'll be talking to you about the Lab Inventory Management System, or LIMS. The LIMS has been developed by the data team, led by myself, Shannon Albeck. LIMS contributes to many of the activities within this project. Wherever samples are collected, we aim to track those samples and store the resulting information from lab analyses. We've made great progress, especially in the last 6 months, where we have developed much of the code, and we're now working on implementing that code so it can be used by our team members. Now we will show you a short video describing how LIMS works.
Hi there, this is Alex Berkeley again to tell you about a project-related facility, in this case the Advanced Research Computing Center. This is an existing facility on campus. The timing of our grant project beginning aligned nicely with an upgrade that was being made to the system. We were able to take $750,000 from our cost share to upgrade and contribute to other resources that were going into upgrading the Teton system. This system is one that we utilize really heavily in this project. We use hundreds of thousands of CPU hours each year. With the system, we're able to do large-scale computation. We're able to do analyses of simulations, run lots of simulations, do statistical analyses of those. We do all of our manipulation of genomics data in this system, including working with large files that we obtain from sequencing centers, or the large number of files that we obtain when we then split those large sequence files up. This resource is really critical to us accomplishing work that we wouldn't be able to do on desktop systems, and ensuring accessibility of our data to all members of the project simultaneously. Going forward, we'll also invest $50,000 in shared storage for the system, and shared storage that will go within the Advanced Research Computing Center. The shared computing facility is within what's called a data center on our campus. This is a really top-flight data center with high-quality networking, high-quality conditioning of the internal environment. We have chilled water that cools these rack systems, and a really good physical plant that supports the work in this system. As you can see, these racks are not conventional in that they do not take in fresh air on the front and exhaust it in the back, but as I said, they're air-cooled. We have really great networking within the cabinets that includes both conventional Ethernet connections over copper, but also fiber optic connections amongst all the nodes in the high performance storage within the racks. The facility is staffed by IT personnel who maintain the physical environment and the performance of the systems and that surround the racks. The individual racks and computers are managed by staff who report to the Office of Research and Economic Development. In terms of sustainability of this investment in a research facility, this facility and the staff are sustained by the annual budget of the Office of Research and Economic Development at the university and are in the budgets going forward in time. Importantly, usage of this facility is open and free to all members of the university community, whether it be students, postdocs, faculty, and whether or not those members have invested in the shared facility or not. We see this as a really critical way of approaching the shared facility so as to stimulate competitive research across campus.